article, the first thing that proteins do is they act as enzymes. Now, that's just the name of the thing. That's not really an explanation. Enzymes are anything that speeds up a chemical reaction. So if you said, hey, chemical reactions would happen slower if I didn't have proteins, you would be correct because they are required to act as enzymes. Now, if you worded this differently and you said your metabolism would decrease, that would definitely be something that would happen as well. Since enzymes help break down the food that you eat, you could say that you had problems digesting because an enzyme helps with that. Anything in that area would be something to do with proteins acting as enzymes. Now, the next thing on the list is about structure. And so there's a couple that are mentioned specifically: keratin, elastin, and collagen. Maybe the guys not so much, but lots of female type lotions and products have especially collagen in them if it's lotion. Keratin you often find in fancy shampoos. So those are substances that are there for structure. So I know I always think about hair and nails and skin, and the fact that since proteins help contribute to their structure, the quality of your hair, nails, and skin would probably decrease. Now, there are other types of structural proteins. It's not just those ones. Proteins are part of the structure of basically every system in your body. So if you gave an example of an organ in your body that would not have as good of a structure because proteins were lacking, that would be acceptable. Storage is the next one. For example, iron was the one given in the article. There's also the storage proteins found in eggs, egg whites, breast milk, anything that would feed an unborn fetus or an unborn baby. Those are all areas where protein storage, where protein stores energy, or where protein stores minerals like iron. So if you said you might lack iron, or if you said that you might lack energy because of protein storage, that would be an acceptable answer. Transport is the next one. Usually the example that people can think of is the one that has oxygen, where there's a protein called hemoglobin that transports oxygen around your body. If you lacked protein, you would probably lack hemoglobin and thus lack oxygen. That would make you feel tired all the time. The next thing on the list is that proteins can act as hormones. Now, the one that's specifically mentioned here is insulin. So if your body lacked protein, you might not be able to regulate your blood sugar. Now, the next thing on the list is receptor. So proteins are part of the cell membrane. That's something that you probably remember from Science 10. And they help regulate what goes in and what comes out of a cell. So if you said anything about things getting in and out of cells, that would be a thing that proteins normally do, and that might be lacking if your diet lacked protein. The next one is contractile. And this really has to do with muscles. There are proteins that your muscles require to perform contractions, and if you don't have them or if you have fewer of them, it will be difficult for your muscles to contract. The examples there, you could have said myosin, actin, kinesin, dynein. They're all proteins that help with contraction of muscles. The last thing is defense. Proteins are what make antibodies. Antibodies are the specialized structures that will attach to foreign objects in your body, foreign cells, foreign organisms, so that your body can attack them and get rid of them. So those are the 8 things that are on the list. And so that you know, we are going to talk about virtually every single one of these again as we go through the chapters that we're going to do, the first 10 chapters of your biology book. So this is the continuation of lesson 4. We just summarized the 8 functions of proteins. So the next thing on our list is how can we affect the structure and function of a protein. And there's 2 things here which I'll talk about in turn. The first thing I could do to a protein that could affect their structure and their function is to denature them. So denature, that's the verb. If I wanted the noun, I would call it denaturation. So when I denature a protein, this is when proteins are temporarily, and that's a key word, temporarily changed by physical or chemical processes. Now, what does that mean, a physical or chemical process? Something physical could be just a change in temperature. A chemical process could be something like adding an acid that affects the pH. Now, temperature and pH would be the 2 things that I would use as part of a question if I were to ask you about it. So to denature a protein is when they are temporarily changed by physical or chemical processes. Now, since we're focusing on enzymes, I'm just going to remind you, enzymes are proteins. So proteins or enzymes are denatured by mild temperature or pH. Mild would be another key word. Now, everything that I've highlighted here is trying to emphasize that this is not a permanent process. This change is definitely reversible. So on the right, there's a picture that's trying to illustrate to you what it means to denature a protein. On the left, you have a normal protein. And so that you know, proteins are not just a linear chain of amino acids. Proteins are amino acids linked together, but then I take this strand and it gets all wound up and curled around and folded over itself. And that structure helps give proteins their function. So it's true. A big long chain of amino acids can make a protein, but it's a little bit more than that. A protein structure depends on how the chains of amino acids are folded over each other or wrapped around each other till we get a 3-dimensional structure. So normal proteins are in 3D. When you denature a protein, so that's the picture that's on the right, it kind of looks unraveled. Like I've undone some of the twirling up, like I've stretched it out, like I'm turning it back into just a long strand. Now, a really good analogy for this, which you might not even be familiar with because it's so old-fashioned, is a cassette tape. Now, when I was growing up, cassette tapes were cool. They were the new 8-track, and it was very awesome if you had a cassette tape player. Same thing with VHS videotapes. You probably don't even know what those are. But there's an actual piece of tape inside of them. And something that used to happen is that every once in a while, your tape player would start pulling the tape out of the cassette holder. And this is exactly what it would look like. So the tape would come out of your cassette tape, and it would be all unraveled. All you had to do to fix it was take a pencil, stick the pencil in the little groove, and twist it back up. So I could accomplish renaturation. Renaturation is when I twist the protein back up into its shape, or in my cassette player analogy, I twist the little gear and the tape gets rolled back up inside the cassette. And the key thing here is I could do that a number of times. It could get pulled out, I could twirl it back up again. So denaturation is reversible, it's not permanent. Now when there is a permanent change, we would call that coagulation. Now coagulation is when proteins are permanently changed by the same kinds of chemical or physical processes. So you'll notice I mentioned heat again, and the same thing would be true with extreme pH. In denaturation, we said that mild temperature or mild pH could cause it. If temperature now becomes excessive or pH becomes excessive, proteins will melt. That'll be kind of what it looks like from the outside, but they'll break apart. If we talk about frying my egg over here, I can't uncook the egg because my egg has now coagulated. The proteins that are inside of it have been permanently affected by heat, and so I cannot simply wind them back up and regain the original structure. The structure is permanently altered, permanently changed. Does anyone have any questions they'd like to ask about either of these two processes? Yes. And usually the difference between whether they're going to denature or coagulate is how excessive the temperature is. Now we'll talk about, probably we won't get there today, but on Monday, that different proteins have different optimum ranges. So you'll notice I didn't give an actual number here, and that's because some proteins actually like a really low pH. So for example, you have proteins in your stomach. Your stomach is really acidic. If those proteins were going to coagulate, they wouldn't function in your stomach. So there'll be some proteins or some enzymes that can handle a lot of heat or a really low pH, and others that can handle a different number. So these definitions are really general on purpose, and it's because every protein has a different optimum range. Does anyone else have any questions they'd like to ask here? So then we're going to start talking about enzymes. Now you'll notice that there is a picture on the side there, and I have a number of different versions of pictures of enzymes. I'm going to highlight the part of the picture that I feel is the most important, so perhaps if you feel like art is not your thing, you can pick out the big important thing. So that we know all enzymes are made of proteins. Not all proteins are enzymes. Proteins can do 8 different things, but enzymes are all protein. There are over 1,000 of them, so obviously I'm not asking you to know them all or name them all. We will pick a select few that are directly related to the processes that we're going to discuss. And for right now, we're just going to talk very generally about enzymes rather than name them all up front. The way that you can recognize an enzyme most of the time is that a lot of enzyme names end with ASE. Not all of them, because it's science, so there's always exceptions to every single thing, but a lot of enzyme names end with ASE. In fact, if you were reading something in this science class and it was a word you didn't recognize but it ended with ASE, I would say 99% of the time that word is an enzyme. Now what you'll see is that for most enzymes, ASE is the ending and the beginning of the name is the substrate or the substance that they're actually going to break down. So just as an example, I'll let you know that sucrase, which is the enzyme I'll use here as my example, sucrase breaks down sucrose. Makes sense. So we talked about disaccharides yesterday. Sucrose is one of them. And the enzyme that breaks down sucrose is called sucrase. And most enzymes have a name that's like that. Maltase breaks down maltose. Lactase breaks down lactose. It's usually named quite conveniently. There are a few that don't follow that pattern, and when we get to talking about them, I'll make sure to point it out. Now, what's the job of an enzyme? They are a catalyst. Now, there are other things that can act as catalysts. Enzymes are one example of a catalyst. A catalyst is something that speeds up chemical reactions but is not altered and can be used over and over and over again. So an enzyme isn't an actual reactant or product in a chemical reaction. It's there, it's hanging around, it's in the neighborhood, but it does not get changed because of the reaction. This means it can be used over and over and over again. And the conclusion we would draw from that is that you don't actually need a lot of an enzyme because I can use it over and over and over again. Now, the key thing about catalysts and enzymes in particular is that they will accelerate the rate of a chemical reaction that already occurs. So they're not making something happen that wouldn't normally happen. They're making something happen faster. So they cannot make a reaction occur. They can make it happen faster than it normally would, but they can't make it happen. So on the right, I have a picture of sucrase, and it's a very generalized picture. You'll see that there's a big 3D looking glob thing. That's my protein or my enzyme. And what I want you to notice is there's a spot on the protein. Now in this particular picture, I kind of feel like this looks like a ghost when I look at it. It's like a weird shaped thing with two eyes. That is called the active site. It's the place that the molecule that's going to be affected is going to attach. Now over here, I have sucrose, and it is called the substrate. So I have an enzyme, I have a substrate, which is the substance that's being affected, and I have an active site. And the idea is that my substrate, like it is illustrated here, will go and fit itself into the active site. And now here's the key thing. The enzyme's not doing the reaction. The enzyme is simply making the reaction possible. This is a molecule of water. Water is going to join up and cause there to be a break between the two pieces, the two subunits of sucrose. We know that this process is called hydrolysis. So sucrase doesn't make hydrolysis happen, it just makes it happen faster. It provides like a meeting spot. If you want, you could think of an enzyme as being kind of like a matchmaker. It's like, hey, I think that you would go really well with water. Why don't you come with me? I'll help you find a molecule of water. We'll put you together. So water and sucrose are the two things that are actually part of the reaction. The enzyme is the place that it's happening. At the end then, I have glucose and fructose as my products. So the enzyme helped a disaccharide be broken down into two monosaccharides. Now we're generally going to talk about enzymes that break things down so that you know it can happen the other way. Enzymes can help things go together, but we're usually going to be talking about enzymes that are breaking things down. Does anyone want to ask any questions up till here? So in this model, we're saying that the enzyme is the key and the substrate is the lock. So the analogy we're trying to make is that the enzyme can unlock the lock, but the enzyme cannot open the door. It simply makes it easier for someone to open a door. So the enzyme is the key and the substrate is the lock. So there are a few things that we need to know about how an enzyme might unlock the substrate, make it easier for the substrate to react with something else. First, in order to catalyze a reaction, the enzyme has to physically come into contact with the reactant molecules. And you might think that that is a point that you shouldn't even have to mention, but one of the things that could help you increase a reaction rate is to create more surface area, to have more physical space for a reaction to happen. And so coming into contact with the reactant molecules is important. So I'll just note that surface area is really key. There needs to be a lot of surface on whatever substance you're trying to react for the enzymes to react with it, to come into contact with it. Once the enzyme has come into contact with whichever molecule is reacting, it is called the enzyme substrate complex. Now, like you saw either in the video or on the previous slide, the substrate, which is the reacting molecule, the thing that I'm trying to break down, attaches to the enzyme at the active site. And that's the spot that is the correct shape. It looks like it's a little puzzle, and the substrate fits exactly in that spot. Now, I alluded to this, but the substrate could be broken up. That's called catabolism. Or the substrate could be attached to another substrate. That's called anabolism. Some of you might be familiar with the term anabolic steroid. It's the kind that you're not supposed to use, but that people would use to build up muscle mass. And that's because anabolism is to build something up, whereas catabolism is to break something down. And then, like I mentioned in the video, enzymes have very high substrate specificity. That means that they won't attach to just anything. They will attach to one type of compound. And this all depends on the shape of the active site. Now you'll see I circled the word type, and I didn't on purpose write the word they'll only attach to one compound or one substance, because that's not actually true. Most enzymes will attach to one type of compound. That means that another compound that's similar could probably attach to the enzyme as well. That could be harmful or it could be helpful depending on what the substance is. So just like putting a key in a lock depends on the shape of the key and the shape of the lock, a substrate attaching to an enzyme depends on the shape of the active site and the shape of the key. Now, if you've had the unfortunate luck that I have, you might be aware that lots of keys will fit into a lot of locks, but not necessarily unlock them. And that's what we're talking about here with substrate specificity and whether or not we can attach to one or more than one type of compound. I could put a lot of different keys in the lock. Most of them will just block it and not let it open. Only a particular shape will actually function in the lock. So there's an illustration there that shows the same thing as the last page, but just in a different format. And it adds one particular thing. This idea right here. So down at the bottom, where it says, how do enzymes work? It says that the presence of an enzyme will speed up the reaction because the active site will facilitate the chemical change. In other words, we're going to lower the activation energy. Lower the activation energy. That means we're reducing the energy required to start this particular chemical reaction. So we need energy to make or to break a bond, and you always need a bit of energy to get started. And so what does the enzyme do? It weakens the bonds in the substrate, if we're doing catabolism, by lowering the activation energy, making it easier for this reaction to happen by reducing the amount of energy we need to start the reaction off.